I'm Dr. Michaela Patterson from the Medical College of Wisconsin, and I just moderated a session on cardiac development. And I'm here with one of the speakers, Dr. Muge Martinez, who gave a fantastic talk about RBFOX and hypoplastic left heart syndrome. And I was wondering if you could tell us a little bit about it.
Yes, yeah, I actually presented some of our work related to RNA binding protein RBFOX2, which has mutations in patients with HLHS, and we generated a knockout mouse model, deleting it specifically in endocardial and myocardial cells, and found that these embryos did not develop four-chambered heart, had problems in outflow of tract, as well as vasculature development.
Yeah, so RBFOX2 is an RNA binding protein. So can you tell us a little bit about some of the mechanisms you guys have discovered related to how it's acting?
Yes, so RBFOX2 is an RNA binding protein that's known to regulate alternative splicing in many different cell types. And we found that RBFOX2 regulates Rho GTPase cycling genes, which are important for cell adhesion and proliferation. And we were able to show that we can switch the splicing of these genes, and then this splicing switch was adequate to change the cell cycle as well as cell adhesion phenotype in endothelial cells.
And then the polyadenylation phenotype?
Yes, so we also found that we have recently shown that RBFOX2 also regulates alternative polyadenylation of pre-mRNAs, which can affect 3' untranslated region length. And we found that in the embryonic hearts, RBFOX2 can regulate the 3' UTR length of genes that are important for metabolism, specifically some of the very important mitochondrial genes implicated in cardiomyopathy.
Are the polyadenylation defects happening in a specific cell type to the best of your knowledge?
We don't know exactly, and that's some of the things we're doing specifically, cell-specific deletions and cell-specific changes in splicing and polyadenylation, whether they differ. These are open questions.
So are you finding that any of these defects you're finding in the mice recapitulate what you're seeing in babies with HLHS?
So it's been reported that the mitochondria is affected in HLHS patients, as well as some cell adhesion genes have been reported to be affected in these patients. So it really fits to some of the phenotype that's seen in the patients with HLHS.
So do you have any last thoughts of where you're going with this beyond the cell type specificity questions you're asking?
Yeah, we're very interested in to identify RBFOX2 specific targets in the heart, and that we are doing some experiments related to that using mouse embryo hearts, which is extremely challenging, technically difficult, but we are very excited about the direction.
Wonderful. Thank you so much for talking to me today. It was really exciting work.
Thank you so much. Appreciate it.